doing in the University of Vigo for some time. So all of us know that sequencing a genome is now something quite affordable. Almost all of us can do it. So the thing is, its cost is cheap. So still the amount of information that you get from sequencing a whole genome is kind of astonishing. So the ECMG did an amazing work providing us with the tools to classify the mutations into pathogenic, likely pathogenic, variants of uncertain significance, likely benign and benign. But still, we have issues, especially with those in the middle, with likely pathogenic, VUS, and likely benign. Here is where functional studies play a major role into pushing these mutations into either pathogenic or benign. So if you think of a gene, like this scheme here, and I ask you about mutations, most of you will tell me, of course, exonic, they are the disease-causing ones. But there's plenty of more places where a mutation can happen, and all of them are interesting, like introns, splice sites, and my personal favorite, promoter regions and UTRs. Given the genetic framework of PAH, we have plenty of proteins with different roles, a lot very varied receptors, transcription factors, and also potassium channels. So if you want to perform functional studies, you have to take into account the role of the protein you're screening for. You can't treat the same way a transcription factor like TBX4 or a potassium channel like ABCC8. So today I will be showing data from ABCC8 and TBX4 from all of them from mutations of the Spanish cohort. So for splicing variants, we use the PSPL3 vector, which is an exon trapping vector. This means that we can insert between two artificial exons our inserts, which consist in intron, exon, and intron from the patient or from a wild-type donor that we then mutate into getting the mutated genotype that we want. So in this case, for example, we got exon 27 for ABCC8 gene, and this mutation here wasn't actually a splice site mutation. It was a missense, or it was predicted to be a missense by the in silico tools. So we carried the mini gene, and we got a total exon skipping, which is way worse than a missense. But we can do way more with the PSPL3 vector, because sometimes exons are processed as packs. So when we saw this mutation in the splice site of ABCC8, I mean from TBX4, we thought, well, maybe the splice is only exon 7, or it also does it with exon 6. So we cloned it, we did the mini gene, and we got a double exon skipping, which is something quite unusual. But I want to speak about other type of mutations, like non-coding variants, but in other places. In this case, promoters or UTR regions. So we have been working with the endothelin gene, which is not a causal gene for PAH, but rather than, it may be only a, I can't recall the name, so it may be a modifier only. So I've been working with Carlos Lopez Solarat from our lab, and we first screened the promoters and found two common SNPs. And after an in silico analysis, we found that maybe these SNPs were tweaking a bit with the binding of some transcription factors like KLF4, PPARG in the 5' and VDR in 3', the vitamin D receptor. So we prepared luciferase plasmids, we inserted the UTR regions and the mutations, we had all possible genotypes, and we did the luciferase. What we got is that these common SNPs that were found in PH patients were actually increasing the amount of luciferase that we were finding in our assay. This could probably correlate with an increase in endothelin, but we're currently validating it using knockdown models and luciferase again, and well, Western blot and a lot of other things. But I want to really speak about missense variant because, yeah, of course, they are the disease-causing ones usually, but they can be tricky to analyze. So I did a research stay at Nick Morrow's lab in Cambridge last spring, and we started a project where we are currently analyzing all the TBX4 missense mutations from the Spanish cohort and the UK cohort. So for now, what we got is a model that responds to TBX4 in a dose-dependent manner. You can see there from the unstimulated to up to 100 nanograms. It increases the signal, but the interesting bit is this, because we have 200 nanograms there of the wild type, and we have the same amount for a terminus mutation right in the middle of TBX4, which is no protein. And then we have another mutation from the Spanish cohort that was characterized and classified as a likely pathogenic variant, but with this assay, we see that it has the same effect as the null control that we used, so we pushed it to pathogenic. We now have the data of most of the variants. We have done the mutagenesis for most of them, and we will have all the data pretty soon. But we don't only do functional studies, we also try to do translational research. So we got plasma samples from healthy controls that were given to us by the OnComet group in Santiago de Compostela, and also from PH patients of functional classes 1 and 2. We got them from the Servicio Galego de Saúde, from our collaborators, and also from the clinic, from Dr. Barbera and Dr. Isabel Blanco. So we extracted myRNAs, did RNA sequencing, and then a differential expression analysis. We found 26 different myRNAs that were expressed differentially between the patients and the controls, some of them really interesting, functionally speaking. So we did this analyzing using two tools completely different, which are DSEQ2 and DETGR, and they showed us almost the same results in fold change and also FDR, which we use at 1%, which is really low. This kind of makes it quite robust. So this is the heat map that we got from our differential expression analysis. And you can see here a group of PAH patients that is always clustering together, while there are other groups that are actually right in the middle with the controls, or a bit more to the left, I mean to the right. So we're currently working with this because we really want to know what is happening here, why are they clustering that way. So now we are doing classification models, and we are trying to get very sensitive myRNA panels to use or aid in diagnosis, but well, we may be late, as Richard said before, so it's something good to know. Okay, so we are currently recruiting our validation cohort, and we need around 120 PH patients, the same number of controls, and between 20 and 30 patients of other pulmonary vascular diseases. So we're working on it. We're trying to validate our myRNAs using droplet digital PCR, which is a state-of-the-art technology for the quantification of these myRNAs. So at the moment, we got funding from Actelion and the Xunta de Galicia, but we're looking for new sources to keep this up. So what I want you to take home is that functional analysis can help into pushing mutations from likely pathogenic, likely benign, and VUS into either pathogenic or benign. It's kind of compulsory sometimes. Also, non-coding variation in PAH is important. And of course, that myRNAs may be of aid into diagnosis PAH. So I would like to thank the organization again, and all my collaborators that are here today, and well, all the people here, which were pretty important to us. So thank you.
Thank you very much again for being on time. So, questions from the hall?
Mauro, una pregunta muy chula la charla. De las muestras que habéis incluido para hacer estudios de microRNAs, de los tipos de hipertensión pulmonar, ¿habéis visto que se clustericen igualmente los que tienen el mismo subtipo o etiología de hipertensión pulmonar? ¿O cómo los habéis incluido para hacer microRNAs? Y si habéis visto alguna correlación, o realmente esto sirve para ver que se correlacionan probablemente diferente a nivel molecular de clínico.
Okay, so there's a couple of things. First of all, we got only IPAH patients but we didn't do any genetic diagnosis on them. We want to imitate the clinic, how the clinic goes. I mean, they get the patient, they don't know what it has. It can be PAH, so we wanted to try that. But we sent the samples for genetic testing. I think you have all of them. And some of them were actually from the rehab. So if we need the genetic data, we will ask for it, and we will try to see if the cluster is explained by BMPR2 mutations or TBX4 or something else.
Final question.
Thank you for the talk. Regarding the functional analysis, do you go a bit further than just knowing what the consequence of the variant? I mean, do you go further, like expression analysis, or what is the meaning of this lower expression or higher expression, or just?
So it depends. Sometimes we can't really. I mean, the ideal thing would be to work with VOECs from the patients. I know how to extract them, but it's kind of tricky to get them because you have to get the samples quite fast. So ideally, I would love to get the patients, extract VOECs, and work with them functionally to know what is happening downstream in the cascade. But usually we try to use knockdown models with VAECs, primary or smooth muscle cells. We do what we can, to be honest.
You have mentioned that about the work you've done with 2 non-coding regions, UTR regions,
Yeah.
and the question is there an interest, and how would you escalate this kind of analysis to other genes rather than these 2?
To other genes, if you can do a whole genome, I will try to do that.